Hello everyone. We're here at Cold Spring Harbor Laboratory for the 86th Cold Spring Harbor Symposium on Quantitative Biology. The topic of this year's symposium is genome stability and integrity. My name is Peter Sherwood. I'm a molecular biologist, and I'm joined today by Agata Smogorzewska, who is head of the Laboratory of Genome Maintenance at the Rockefeller University in New York. Agata will be presenting her work later this week during the symposium, but I'm happy to welcome her here today now.
Thanks. It's great to be here.
Super. Maybe I'll just give a minute of what I see as what you're working on, and then you can correct everything. Our cells in our body are constantly under assault from external agents as well as internal agents. So externally, sunlight, smoking, obviously, drinking. But also internally, there's natural biological, biochemical processes that generate things that damage our DNA. So
That's
DNA
right.
repair is crucial, and when it goes wrong, there can be a big problem. So perhaps you'd like to tell us about your work and a little bit of background, and then go into your current results.
Sure. Yes, so you're definitely correct, and my lab is interested in how this exogenous and especially endogenous DNA damage is repaired. And we are interested in how it's done during DNA replication. And we know that this is very important by learning from human diseases. So we study this particular rare disease called Fanconi anemia. And I think of the disease as a disease of stem cells. It turns out that patients with Fanconi anemia develop bone marrow failure, so they are unable to make red blood cells, white blood cells, platelets. They also have problems with germline, so they're subfertile or infertile. And then and I forgot to say, they also can have a lot of developmental abnormalities. So during development, the stem cells are somehow vulnerable without this repair pathway. And a major focus of our lab right now is understanding why and how these patients develop tumors. So these patients are very much prone to head and neck squamous cell carcinoma and also anogenital squamous cell carcinoma. And until now, we really didn't understand how they developed these tumors and why they were so susceptible to the tumorigenesis.
Right. So
maybe I'll tell you a little bit about the actual lesions that the Fanconi pathway repairs, because that becomes very important, and it ties with something that you said about smoking and metabolites in our cells. Mm-hmm. So the Fanconi pathway is a very complex pathway of DNA repair. I already mentioned that it repairs DNA during replication, and it repairs a very particular lesion that's called an interstrand crosslink. And this is a lesion that covalently links the two strands of the DNA, the Watson and the Crick strands. And the repair of this lesion is imperative because you need to separate the two strands of the DNA for replication and transcription, and you really cannot maintain the genome if you cannot separate the two strands.
Right.
So in Fanconi patients, that repair doesn't happen normally, and that creates double-strand breaks, and it happens and creates genome instability. And the other aspect that's interesting is that we believe that the endogenous DNA damage is really creating, or endogenous metabolism is creating a lot of the DNA damage that the pathway repairs.
Right, even in normal people that don't have Fanconi, it's happening all the time.
That's right.
And
That's
normally
right.
It can be repaired and everything's fine, but
Right.
this disease has highlighted what the problem is, how this pathway does work when it works,
That's
and
right.
in this case when it doesn't work, what goes wrong.
That's right. And we believe that from work from others like KJ Patel, who's shown that in the bone marrow you really need first to detoxify some of these chemicals like acetaldehyde and formaldehyde. But even if you have normal detoxification, systems, you still have a little bit of these chemicals that can get to the DNA, and because they're very active and they can react with the DNA, they make these cross-links.
Right. So I know that in your current work that I think you're going to be talking about later this week, you identified what happens to the genome rearrangements and oncogene amplification and
That's
other
right.
things that happen. Maybe you could tell
That's
us
right.
about that.
Right. So since we didn't know really anything about the tumorigenesis process in Fanconi anemia patients, and we didn't know whether it was similar or dissimilar from the sporadic cancers that occur in the head and neck in patients, we took an approach of studying the tumors themselves. So I'm a head of International Fanconi anemia Registry at Rockefeller, and we've been collecting tumors for a while now, and we also had some archival tissue that's been collected in the past. So our approach was to take these tumors and do sequencing, a whole exome, whole genome. We've done some spatial Visium sequencing. We've done some single cell sequencing, although we have to do much more of that to understand other aspects of the disease that we haven't yet studied. And by looking at the genome, we could figure out what was the main kind of lesion that led to tumorigenesis.
Right, and what did you find?
So we found that that a lot of the changes that occur are what we call structural variants. So these are changes that reshuffle the genome, and these can be deletions, they can be inversions, translocations, some tandem duplications. And what's striking is that in Fanconi anemia patient tumors, the numbers of these lesions is very high.
Higher than you see in other tumors.
That's right. So it's about 2 to 3-fold higher than in sporadic head and neck cancers.
Okay.
And it's also, it's at the same level as seen in, for example, BRCA2 mutant breast cancer or ovarian cancer. Right. So very high levels of structural variants. And what's interesting is that they occur throughout the genome, but during the tumor evolution, we can see that certain changes are selected for. So the tumors are full of deletions of tumor suppressors, like P53, and amplifications of a lot of oncogenes. So these would be MYC, PIK3CA, cyclin D1. And what's also interesting is that unlike in sporadic cancers where there are some structural changes, and we can return to that, a lot of changes, a lot of drivers are mutated by point mutations. In Fanconi, it's really the only gene that's mutated by point mutations is P53, and we think that that's an early event. Right. And then that mutation is followed by allows this huge genome instability to propagate. So all of these structural variants are arising in the setting of P53 mutation.
I see. Are you, what are your next steps going to be? Are you considering therapeutic approaches that maybe have come out of this work?
Right. So from looking at the genomic landscape of these tumors, we can definitely suggest that one should be thinking about multi-pronged approaches, because there are so many different pathways that are affected in these tumors that just going after one of these definitely will not solve the problem and will not lead to better outcomes. So you would have to, let's say, inhibit PIK3CA at the MYC at the same time. It's easier said than done, but try to have multi-pronged approach. The other aspect of what we are very interested in doing is to identify what are the endogenous sources of this damage in Fanconi patients, in keratinocytes specifically. So we believe that each cell in the body will have specific metabolism pathways that are active that will create specific chemicals,
right.
and that those chemicals will create a damage that now will specifically require one DNA repair pathway and not another. And for Fanconi, these are aldehydes, and maybe some other as well, but the aldehydes are really important. We know that.
Right.
But we think that there will be specific aldehydes in the keratinocytes, so that's what we are trying to identify.
In normal cancer patients that don't have a syndrome like Fanconi,
well,
there would be a particular kind of aldehyde in their squamous cells.
Well, so we are actually the key is really to understand the normal keratinocyte biology, because we think that especially in Fanconi anemia patients who develop these tumors on average at 30 years old, or even as teenagers, and not all of them have exposures like to tobacco or to alcohol, that this endogenous DNA damage, if decreased, might move the progression to tumorigenesis to a later time. In sporadic cancers, we think that the main culprit, although the endogenous aldehydes are not helping, but the main culprit is the tobacco smoke and the alcohol.
I see.
And the reason why we think that is because in sporadic cancers, we see, as I said, there are some point mutations, like in PIK3CA and others in other genes, but that cancer is also full of structural variants, not as high numbers as in Fanconi anemia, but you still see a high percentage of structural variants.
So it's a signature of this type of damage, the aldehyde damage that we're talking about.
That's right. So we tried to make the connection that in sporadic cancers, even though in those patients, the Fanconi anemia pathway is wild type, so it's genetically unaltered. The sheer number of these damage and of these lesions coming from tobacco smoke, for example, now overburdens the Fanconi anemia pathway. So even though you have normal levels, it's not enough to keep the genome stable, right?
Yeah.
And we don't know exactly why and what's sort of the key step that might be inefficient or not sufficient for the repair, something that we're interested in learning about.
Yeah.
But we see that there is a correlation between structural variant numbers and tobacco history, smoking history.
Right. This gives me the idea, and maybe we can close on this, which is that there are long-term smokers, don't smoke people, but there are long-term smokers that survive. Is it possible that they have some sort of supercharged
DNA repair pathway?
pathways?
Yes, it's certainly possible.
And
making a more efficient DNA repair, stimulating DNA repair might be some way of prevention. But some of it might be just luck and just accumulated damage and some other mechanisms, right? So another issue in Fanconi patients is that their immune system might not be perfectly normal. And in sporadic cancers, we think that immune system obviously has a role in suppressing tumorigenesis as well, and so there are a lot of variables when we are thinking about smokers, but in general, it's not a good idea to increase your DNA damage to the point of tumorigenesis.
Right. Well, thank you.
Yeah.
It's just fascinating the way that you've used this syndrome to uncover sort of the normal biology that underlies so many things. So thank you for coming.
Thank you so much.
See you later.
Thanks.
Bye.